Alright, so the last segment of our discussion is on the process of translation. Alright, so here's a short animation kind of giving you an overview of the process of translation.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into three stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anti-codon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anti-codon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Alright, so looking at this process of translation in a little bit more detail. All right, so you have messenger RNA, which we've already talked about from our previous discussion on transcription. Now you also have another type of RNA called transfer RNA. The function of transfer RNA is that it transports amino acids to the ribosome. In this case, these amino acids are going to be used to assemble your polypeptide chain, or your protein. Now, your transfer RNA. You have many different types of transfer RNA that will bind to a specific amino acid. Now, on the stem loop structure of your transfer RNA, at the base of your transfer RNA molecule, you have the stem loop structure. It forms a cloverleaf pattern, and at the very bottom of it, you have a sequence of RNA nucleotides that will be complementary to the codons in your mRNA transcript. Alright, so they'll pair up. And the codon in your messenger RNA, you know that this encodes for a specific amino acid, right? Well, that anticodon on the transfer RNA will have that specific amino acid attached to that tRNA molecule. Alright, so here, this is showing you the tRNA molecule here. Alright, so you have this nice cloverleaf pattern, alright, stem loop structure, and basically RNA is capable of forming this type of pattern because the RNA molecule itself, it's one linear chain of RNA nucleotides. Right? And you can have hydrogen bonding within the RNA molecule itself. So it can conform to this basically secondary structure, if you will. And at the base of this tRNA, you have a sequence of 3 bases that make up the anticodon. Alright, so the anticodon is complementary to the codon found in the messenger RNA. And then you're going to have an amino acid that's attached here on the 3' end of your tRNA molecule. That amino acid will correspond to the amino acid designated by the codon in the messenger RNA. Now, within the genome, you have genes that correspond to transfer RNA molecules. Now, these transfer RNA molecules, once they're synthesized, they have to be loaded with their specific amino acid. Alright, and there's a particular enzyme called amino acyl tRNA synthetase. Alright, and this enzyme is involved in attaching that specific amino acid to the tRNA molecule. Now, before, I talked about how when you're looking at the mRNA transcript, and you could have various codon or codons that corresponded to a single amino acid. Alright, because the genetic code is redundant. Well, for instance, you can have this particular codon sequence corresponds to a specific amino acid. And you can alter the third nucleotide in that triplet code to another RNA nucleotide, so like an adenine or a uracil or a guanine, and you get the same amino acid. Alright, so the third nucleotide in a codon is referred to as the wobble base. It's a wobbly base. It can alternate, it can change, but most of the time, not always, but most of the time, you'll get the same amino acid that's encoded for by that codon. Alright, because genetic code is redundant. Alright, so here you have the tRNA molecule. Alright, so this is a tyrosine tRNA molecule. Alright, notice that this specific tRNA molecule has a anticodon sequence of AUA. Alright, and this tRNA molecule will bind to the enzyme tyrosyl tRNA synthetase. Alright, so it's specific to this tRNA molecule. You have the amino acid tyrosine that binds to its active site. This enzyme will bring together the tyrosine and the tRNA molecule so that they can be covalently linked to one another. Alright, so this requires the use of ATP to catalyze this covalent bond that links your amino acid to your tRNA molecule. Once this is done, your tRNA molecule with its respective amino acid is released. At this point in time, this tRNA molecule is referred to as being charged, or it's loaded. Alright? Basically, it's ready to do the task it's supposed to. Alright? It has an amino acid attached to it. It's a charged tRNA molecule. Your ribosomes. Alright, so your ribosomes are the site of protein synthesis. Alright, so this is where your messenger RNA and your tRNA molecules interact. You can think of the ribosome, alright, it's got a small subunit and a large subunit. So think of the ribosome as kind of the docking station for your messenger RNA transcript. Alright, so here, this is showing you a picture of the ribosome. You have your large subunit and your small subunit. Now, the key thing here is that your ribosome, alright, has three specific sites. Alright, they're labeled as E, P, and A. So those previous videos that I showed you did a really good job of describing what happens at the E, the P, and the A site. Alright, the way that I remember these specific sites on the ribosome. Alright, so A, the A site, is where the charged amino acyl tRNA molecule, alright, the charged tRNA molecule binds. Alright, the P site is where your amino acid is going to be linked through a peptide bond to the next amino acid in the chain. Alright, so this involves the peptidyl tRNA synthetase. Alright, so peptidyl transferase enzyme. this particular enzyme is actually a ribosome. Alright, so it's an RNA molecule, and it's not a protein. Alright, the E site, it's the exit site. So this is where the no longer charged tRNA molecule leaves the ribosome. Alright, so here you have a tRNA molecule located on the P site. Alright, and it's basically going to transfer the growing polypeptide chain to this amino acid on the A site. And once that takes place, alright, this particular tRNA molecule will shift over to the E site and will leave because it's no longer charged. And this particular growing polypeptide chain will be attached to this tRNA molecule. It'll shift over to the P site here, and the A site will become vacant for the next tRNA molecule to bind. Alright, so the P site holds the tRNA molecule that carries the growing polypeptide chain. Alright, so this is where the actual transfer of the growing protein chain occurs to the new amino acid. Alright, this is where you have your peptide bond formation. Alright, the A site is basically the attachment site of your charged tRNA molecule. It binds to the next codon sequence, alright, in line. And then the E site is where your uncharged tRNA molecule leaves the ribosome. Alright, so here, this is kind of showing you the anticodon and the codon. Alright, so here you have your messenger RNA transcript, you have your tRNA molecule here, and notice that your first two bases are complementary, C and G, G and C. Now, here, the last, it isn't complementary. So you know that G and U aren't complementary base pairs. So this can actually vary, right? So it's a wobble position. Alright, so looking at the process of transcription and translation. Alright, so you have your coding strand and you have your template strand. Alright, so your template strand is what's actually read by your RNA polymerase. Alright, so your RNA polymerase enzyme comes in and reads this strand of the DNA. Alright, you get your messenger RNA transcript through complementary base pairing. Each codon has a specific tRNA molecule that's specific amino acid loaded onto it. You have your anticodon that interacts with your codon in your messenger RNA, and here you have your specific amino acids that get linked end to end through peptide bonds. Alright, so which of the following types of RNA contains hairpin loops and is involved in carrying amino acids to the ribosome during the process of translation? Well, it's not messenger RNA. Right? It's not regulatory RNA. It's not ribosomal RNA. It's not primer RNA. Alright, in this case, it's transfer RNA. Alright, so the process of translation, alright, very similar to the process of transcription. Alright, you have three types of processes or stages. All right, you have an initiation stage, you have an elongation stage, and then you have a termination stage. All right, so there are protein factors that aid during each of these respective stages. All right, we're not going to discuss those particular factors. All right, we're just going to say they're translational factors that aid in the process of translation. All right, so during this initiation stage, right, what's happening is your messenger RNA, your tRNA, and your small and large ribosomal subunits come together. Now, first, your small ribosomal subunit binds to the messenger RNA. Then, you have your messenger RNA that basically is pulled through the ribosome, or the small subunit. That ribosome will move along the messenger RNA transcript until it reaches the start codon. And the start codon will occupy the P site. Alright, next, your large subunit will come in and dock on your messenger RNA and your small ribosomal subunit. Alright, so here you have your mRNA transcript. It's binding to the small ribosomal subunit. Alright, and this small ribosomal subunit will move along the transcript until it reaches the start codon. Alright, start codon is AUG. Alright, it's always AUG. Now, in this case, that particular start codon encodes for a very specific amino acid. Alright, so at the beginning of every single amino acid, every single protein that's synthesized, alright, the first amino acid will always be methionine. Except when you're talking about bacterial cells, the first amino acid is formal methionine. Alright, now that doesn't mean that every single protein, the first amino acid is always methionine or formal methionine, because proteins at the end of translation can be modified. Okay? But as they're being synthesized, right, the very first amino acid is always going to be methionine or formal methionine. And it's always encoded by the codon sequence in the mRNA AUG. Alright, so this tells you where translation of a protein sequence begins. Alright, so next you have your large ribosomal subunit that docks onto the mRNA, the tRNA, and small ribosomal subunit. Your first tRNA molecule with the methionine is going to occupy the P site. Alright, so that leaves the A site vacant. Alright, so the next codon in the sequence here will be red, and a tRNA molecule with that specific amino acid will dock at the A site. Alright, so during the elongation stage, alright, you're going to have amino acids that are added one at a time. Alright, so recall that amino acids are always attached to the pre-existing C-terminus, or the pre-existing carboxyl end of an amino acid, or growing polypeptide chain. Alright, this involves the formation of peptide bonds between your respective amino acids. Alright, so the process of translation, alright, it's going to occur in the 5' to 3' direction along the messenger RNA. Alright, so the elongation stage, you have this cycle of steps. Alright, so you have the recognition of the codon by your anticodon on your incoming docking tRNA molecule that's charged. You have the formation of a peptide bond between a pre-existing amino acid occupying the P site or a pre-existing polypeptide chain attached to a tRNA molecule occupying the P site. Then you have translocation. So this is where your tRNA molecules are going to shift positions. The tRNA molecule that occupied the P site will occupy the E site. The tRNA molecule that occupied the A site will not occupy the P site. And now the A site is vacant. Alright, so you can kind of think of the messenger RNA molecule is pulled through at the 5' end out of the ribosome. Alright, so it's kind of like one of those tops that you had this long plastic, basically like key, alright, that kind of looks like messenger RNA, and you would pull it through the top, and the top would spin. Alright, that's kind of the idea, except in slow motion. Alright, your messenger RNA is being pulled through one codon at a time. And this new codon sequence will occupy the A site. Alright, so here you have your pre-existing polypeptide chain, alright, attached to your tRNA molecule at the P site. Your new codon sequence on your messenger RNA will be will have complementary base pairing with your charged tRNA molecule, which will occupy the A site. Alright, so next, once your tRNA molecule occupies the A site, you have peptide bond formation between this pre-existing polypeptide chain and this amino acid. Alright, now this polypeptide chain has been transferred to the tRNA molecule that occupies the A site. Alright, so the tRNA molecule that occupies the P site now is uncharged. Alright, so now this is uncharged. Alright, so the next step is going to be the translocation step. Alright, so here you have the shifting, not necessarily the shifting, but this messenger RNA molecule is actually pulled in this direction through the ribosome, one codon length at a time. Alright, so now this tRNA molecule that's uncharged occupies the E site. This tRNA molecule with a growing polypeptide chain occupies the P site. And now the A site is vacant. The next tRNA molecule can come in and bind. The uncharged tRNA molecule exits or leaves the ribosome. Alright, so you have codon recognition, peptide bond formation, translocation. Alright, these steps will repeat until you have a codon sequence in the A site that doesn't encode for any amino acid. In this case, that particular codon sequence is referred to as a stop codon sequence. Alright, so that tells the ribosome that, hey, the polypeptide that you've been synthesizing is complete. There are no more amino acids that need to be added. Alright, so now we've reached the termination phase of translation. Alright, so a stop codon occupies the A site on the ribosome. Alright, so you have what's called a release factor that binds to the A site. When this release factor binds, it basically causes the ribosome to disassemble. Alright, so your large and small subunit break apart. Alright, your messenger RNA falls away, and your polypeptide chain that you've been producing is released from the tRNA molecule. Alright, so everything disassembles. Alright, you can get your protein, which is the final product of this process. Alright, so here you have your growing polypeptide chain attached to the tRNA molecule occupying the P site. You have your stop codon, alright, which is the next codon sequence. You have a protein called a release factor that binds. Next, your polypeptide chain is released. Alright, so this covalent bond is broken, your polypeptide is released, your tRNA molecule leaves, and now your ribosome and your messenger RNA disassembles. Alright, so this is kind of a summary process of the various steps of translation. Alright, so this is one big summary slide.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Alright, so that particular video, if you'll notice, that it talked a lot about processes that take place in eukaryotic transcription and translation. Alright. And it went into a little bit more detail where it started talking about secreted proteins. Alright. So we're only going to focus on looking at what's taking place in transcription, what's taking place in translation. Alright, what are the big events that are taking place? What are the big players that are involved? What are the steps? What's occurring in each step? And then comparing and contrasting transcription and translation between prokaryotic and eukaryotic cells. Like, what are the differences? Okay.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
Alright, so in this case, you can have multiple ribosomes that move along a single mRNA transcript. In this case, you have many ribosomes on the mRNA transcript. You form what's referred to as a polyribosome or polysome. Alright, so in other words, you can make numerous copies of a set of genes that are involved in a chemical pathway, alright, for a bacterial cell, alright, for an operon, very quickly. Alright, so here, this is showing you a polyribosome. Alright, so you have your 5' end of your mRNA, you have your 3' end at the other end of the mRNA. So you have your start, and you have your end of your transcript. You have your incoming RNA subunits that dock on the 5' end, And as they move across and along your messenger RNA transcript, your polypeptide chain elongates until at the very end the protein synthesized. But you can have many ribosomes that are in different phases of translation of that protein, all along that messenger RNA. Alright, so in this case, transcription, translation, and bacterial cells. It's a simultaneous process. Alright, so here, you have your transcript, alright, and you have your growing polypeptide chain. Alright, so here you have your DNA molecule here. Alright, you have your growing messenger RNA here in different phases. Alright. Now as you go in left to right, your transcript, your messenger RNA, is getting longer. And then each of your transcripts are actually being translated into protein. Alright, so the 5' end of your messenger RNA, as it comes off, a ribosome can dock and begin translating a protein. Alright, so these ribosomes on each portion of the transcript are actually in different stages of assembling that protein. Alright, so many proteins can be synthesized from one transcript, as that transcript itself is being written and transcribed. Alright, so here, this is showing you a polyribosome. So you have your messenger RNA here, and you have your ribosomes that are docking. Alright, and they're each in different stages. Now here, look, for this messenger RNA, you have structural gene 1, 2, and 3. And so from one mRNA transcript, you can get 3, alright, in this instance, 3 proteins. And you can have many proteins that get produced because many ribosomes can dock at the starting point of each respective mRNA transcript at the promoter region. Alright, in eukaryotic cells, your nuclear envelope basically segregates the process of transcription from translation. Alright, not to mention that your messenger RNA has to undergo further post-transcriptional modification prior to leaving the nucleus. Alright, so here we have the process of transcription of your respective gene into a RNA transcript. That RNA transcript is a primary immature RNA transcript. It has introns. Those introns have to be cleaved out. Your exons have to be glued together. You have to have the attachment of your 5' cap, your poly A tail. Now your messenger RNA, now that it's mature, can leave the nucleus. It'll then move to the ribosome, where it will interact with the small subunit and then the large. And then you have the production of your respective polypeptide. Alright, so transcription occurs in the nucleus, translation occurs out in the cytosol. And these are two totally separate events. Alright, they're not simultaneous, as you see in bacterial cells.